So there's two things in the Canvas file for this lecture. There is the lecture notes, and then there is basically directions on a protocol of how to do Quickchange. So first, I'm going to walk through what's happening, and then I'm going to literally walk through that protocol so that you guys kind of understand what's going on. So Quickchange. That's just like a nickname for Site Directed Mutagenesis. Okay, so Site Directed obviously means you're choosing a site. You're choosing a site, and you're choosing mutagenesis. You're choosing a site to mutate. So why would you ever want to like mutate something? Yeah, functionality. So when we study the function, we break stuff. We break it, and we often break it by mutating it. So I'll give you an example from my research. Is I studied a protein called SIDB. SIDB is an enzyme that right here in the C-terminus has a protease, a deubiquitinating domain. And in that deubiquitinating domain is a catalytic cysteine. What's unique about cysteines? Yeah, they have a sulfur. And that sulfur is important in the enzymatic function of this DUB. Okay, so it's basically the active site. This is called the active site. And this is actually a really important concept from biochemistry. What's an active site?
It's where the enzyme actually grabs things and pushes them together.
Yeah, it's the thing that you need to like get that activity to happen. So in one case, I was studying SIDB, and I could make it cause sterility in fruit flies. Okay, but we wanted to definitively verify that that sterility was in fact caused by this DUB domain. So in order to prove that, we made a second version of SIDB called SIDB star, in which we made C1025A mutation. What's that mean? What's C1025A mean?
This is being at the 1025.
Exactly. It means you took the cysteine that's normally at position 1025 upstream from the methionine 1, and we mutated it to an alanine. So remember how I told you in the first lecture, alanine is kind of like the vanilla inactive thing. So if we want to kill this enzyme, we make a mutant version of it, and the only thing that's different is a single amino acid that just inactivates the enzyme. It doesn't change the structure, it doesn't change the folding pattern, it just changes whether or not this thing can actually do its job. Okay? And then in this case, the fruit flies that we made were fertile. So we were able to conclude that the sterility up here was specifically due to that DUB enzyme, which had that cysteine. Okay? And then from now, we can do more mechanistic things like what are the targets of that deubiquitinating enzyme, because we know that the sterility is from that specific spot of that protein. So this is a context or a case where you would want to make a mutation. And this is very, very, very common if you need either a positive or a negative control, or you want to specifically study and make a conclusion about functionality of an enzyme. You always have to have a catalytic mutant. Okay, so this is a very critical concept, and we have to deal with this all the time. And sometimes there's other reasons you want to make mutations, so you need to know how to do that. Okay. So how do you do this? So oftentimes, right, you have your plasmid, you have your GeneX. You know you need to make a mutation right here. Okay. So we could do this by SLIM. We could do the whole procedure by SLIM if we wanted. And all we'd have to do is create an overlap on our primer sets that encoded that mutation, right? But SLIM is kind of like the harder way to do it. There's an easier way to do it. It's the same mechanistic concept. All you need to do is so in SLIM there's the 4 primers. Okay. In Quickchange, Quickchange, or again, Site Directed Mutagenesis, but you'll hear me say Quickchange just because that's like the nickname in where the lab I came from. Literally all you do is you make primer 1, primer 2, and they're going in opposite directions, and they overlap like this. So if this is your plasmid, and if this is the site you need to mutate, you make everything match perfectly except that site you want to mutate, you change that, okay? And then it's another protocol, again, where you PCR all the way around the plasmid, and what you're producing are new plasmids that have your mutation, okay? And then the next step, you already should be able to kind of know from SLIM, the next step is we have to figure out a way to select for that mutant version against the template. And how do we do that? Yeah, so there's obviously going to be a DPN1 step, which DPN1 is going to degrade all the methylated template, which your primers were annealing to, so it's going to degrade that template, and all that's going to be left over is your plasmid, which you then transform in. Okay, so that's the basic concept. Now let's walk through the protocol. If I plug that in, it's going to go haywire, so I'm not going to do that. Okay, so let's walk through this protocol. It's going to tell you how to do it, and I've made notes on it. So this is from Rob Tomko. He's a now professor at Florida State University. He's really good. He made this protocol, and this is how I learned to do Quickchange, and it's a very good protocol. So I'm just going to walk through basically what he was doing, and then we'll walk through how to design these primers. It's quite easy. So here's his experimental plan, and this is kind of how I want you to look at this design document and think about when you submit your assignment of your SLIM primers and your Quickchange primers, you want to kind of organize it like this. Like you want to lay out what you're doing, the rationale for what you're doing, what your gene is. Here he has the sequence of his gene that he's targeting. He's got the rationale for how he may design these primers. You want to kind of have that so I can see the rationale. And that way you learn the most too. Okay? So here he's telling you what he wants to do. I'm not going to get into like the details of the biology here, but I'll just simplify. He's studying a gene RPN7. RPN7 is in the proteasome. What's a proteasome? Yeah, so Rob studies proteasomes, and it's a big machine that's made up of many different proteins. And he wanted to know how the proteasome is going to react if he made some mutants, okay? And he has very specific residues that he's interested in. He's talking about 5 mutations here. And we talked about how to know then if you ever see an amino acid number amino acid like this, D95G, D95G, that means he took a D at position 95 and changed it to a glycine, okay? So that's how you annotate mutations that you've made, okay? So he's just kind of walking you through. So he's got his target. His target is going to be RPN7. It's a subunit of the proteasome. I already talked about that. Your target can be anything. Random gene X. Okay, you can choose whatever you want to choose. If you really want to get rigorous, try to choose an enzyme, find that enzyme's active site, and then deactivate that enzyme. Let's see. I said ignore the details here. Basically, the concept is he wants to make mutations so that he can measure growth in yeast to see what happens when you deactivate or mess up the proteasome. So that's the simplified goal. Remember that proteins are little machines. So here he gives you guys don't have to do this, but he gives you a 3D structure. And it's very important to remember this, that proteins are like little machines, okay? And they fold into these 3D conformations with alpha helices and beta sheets. And here he's marked specific sites that he thinks are important, for interacting with particular chemicals or reagents in the cytoplasm, okay? So he's highlighted these, and these are the things he's going to mutate. If we want to change amino acids, we will change the function. I already talked about that. Okay. Okay, so let's talk about how he actually does it. So here again, here's FASTA format. So FASTA format is you get the sequence and you put a little caret going to the right, you usually will have the name of the gene, some identifying stuff. He's saying, so what he's saying here is this is SC. What's SC? Yep, it's the species. So it's Saccharomyces cerevisiae, which is Baker's yeast. So he's saying this is Saccharomyces cerevisiae RPN7. That's the name of the protein. In yeast, when all genomes are sequenced, all the genes normally just get a number. So the original name for this was YPR108W. Okay, and what's this mean? It's on chromosome 16 of the yeast organism. So yeast, I don't know how many chromosomes they have, Dylan? They have 16. So this is on the final chromosome. Good job. So this is on chromosome 16. Okay, and he's got the sequence here. So what are the first things we always want to look for? Look, what's that? That's the start codon. What's this? Okay, exactly. So he's got methionine 1. He's starting it there. We would want to make sure that when we pull off that sequence, we want to make sure it's in the right orientation. So this is 5 to 3 by definition, because 5 will always start with a start codon. Okay, so he's mapped it out here. And look, he has identified, he's telling you, he's looked and he's found, these are the codons that he wants to mutate. So here's a question is, how do you, if you have the thing that you want to mutate, let's see, which one of these is, this is S, so this is the one of these that he's making, S349P. Let's say we know that. How do you find that? How do you find that in this sequence? How do you find the codon for that? Sorry, what? Yeah, so let's see if I can do this. Oh my gosh, I'm gonna have to like plug it in. Let's see. Turn off draw. Select. Oh my gosh. I can do this. I can do this. But I can't right click. How do I cut? How do I copy? I gotta do it. Oh no, I don't. Well, I'll just make it up on the fly. We'll go to XBasy just like May said. Google. I go to this website every day, so start memorizing this. Okay, so let's just type something ATG, 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 ATG. Let's do AAA, GGG. What's a codon? I'm trying to think of what would be like an important codon. I don't know. AGG, TAG. Okay, and then here we can, so imagine you pasted your FASTA sequence in here. We can change the output, okay? So verbose means it's gonna look like this. Let me show you. Come on. Oh my gosh. Okay, so verbose is saying, it's telling you the 3-letter code, methionine, 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 histidine, histidine, methionine, stop, okay? And there's 3 different frames, and there's 3 different frames going the other way, right? Does that make sense? So if we want to find that actual codon, we have to change this to include the nucleotide sequence, and then let me show you what that looks like again. ATG, 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 cat, TAG.
So the different frames are if you start instead of ATG to TG.
Yep. TGA. If you started at TGA, it would be a stop codon. If you started at GAT, it would be a D. So yeah, it's looking at the 3 possible frames. Okay. So now if we ever wanted to find, so in the protocol, what he wants to mutate is S349P. So we would have taken this whole sequence and we would have copied this and we would have put that into XBasy Translate and we would have put show the nucleotide sequence, and then we would find the right frame of reference that we're looking at, And then we would find the specific codon that we want to mutate, and we would copy that sequence, and then we would go to our Word file, and we would hit Control Find, and we would find that sequence, and it should, go back to draw, it should pop up exactly where our mutation is. Does that make sense? So you might initially think, well, to find a codon, I'm going to have to go 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, all the way until you get here. No, do not ever do that. Just find the codon from Expacey Translate, copy paste it, and then Control Find in your Word document. Okay? And that's how you find it. Then once you find it, mark it red. So you mark it, you know this is what you want. Okay? And then what he did is he took a sequence with the thing he wanted to mutate right in the middle. So he just took that, maybe here to here. Okay? He copied that and he pasted that down here. Okay? And he's saying, this is the sequence I'm targeting. This is really the only thing that I care about. Okay? And then he maps out the sense and the antisense. Okay? So here is CTA, he's mapped it out, 5 to 3. He gives the complementary strand. So this is the complement, G matches C, T matches A matches T, G matches C. Okay, and now he's changed the sequence to the mutation he wants. So he's changing TCG to a proline codon, which is CCG. Okay, so he's made that change, and he's also made the change to match in the complementary strand. Okay, now to make his primers. Let's see, clean this up a little bit. Okay, so to make his primers now, there's a couple rules I'll talk about. In Quickchange, I don't worry about annealing temperature at all. One of the reasons is that your template is a purified plasmid. Okay, that's a very, very, very pure template. I'm not really worried about my primers finding stuff in the genome because I'm not using genomic DNA as my template. I'm using purified plasmid. That's pretty much the only thing that's in there. There's some contaminating E. coli genomic DNA, but it's not really an issue. So I don't really worry about annealing temperature, and I don't really worry about non-specific products because the template in Quickchange is always going to be purified plasmid. Okay, CG clamp still applies. So here, if you look at his primers, the forward primer still starts with a C, ends with a G. Here, the reverse primer starts with a G, ends with a G. So CG clamp still applies. You always want to try to find the closest C or G. Okay, so he says I put 4 bases on the 5' end of my desired mutation. So that means you want to start your primer at least past that T. Okay, so he started it here at that C to obey the CG clamp rule. And when you do the quick change, you kind of want it as close to 4 but not less as possible. You don't want this thing you don't want this primer extending further and further to the left because you want these overhangs as big as possible. Those are going to create the sticky ends where this thing matches itself. Okay, so here on this side, here was the rationale. He went 1, 2, 3. I could end with that C, but then I'm not 4 base pairs to the 5' end of my reverse primer, so I got to go further. But if I stop here, I'm not obeying CG clamp, so he goes further, further, further to this G, and that's how the rationale of how he picked that. Okay. And then you extend your primers as far as you can go to the left or to the right on the other side up to 40 base pairs. Okay? So you would copy this here and you could do you can hit review word count or number count or whatever, letter count, character count, okay? And then you make it up to 40 base pairs. And the rationale for this is usually when you buy a Legos, the price for Legos less than 40 base pairs is cheaper than when you buy a Legos that are greater than 40 base pairs. So it's kind of just a cost efficiency thing. You don't go more than 40 base pairs because then you're shooting yourself in the foot with the cost. And you can go down to 35. Okay? So you just kind of want to find something within the range of 35 to 40 that gives you a CG clamp. Okay? And you don't worry about annealing. Okay, let's see. I usually kind of always try to default to 40 base pairs, if possible. Make sure the total and complementarity between a given primer template is greater. Okay, so what he's saying is he makes sure this complementarity to the template right here, these bonds, is longer than here. Okay, so basically the simplified way to say this is make your sticky overhangs greater than your matching, what would you call that? Matching primer to primer. To primer. And that's this region here. Okay? That means here's one of the problems is if you make primers, so imagine the other way to do this. Let's go to the EndNote document. The other way to do this, imagine if you had a plasmid, okay, and you make primers that look like this. One of the problems you're going to get is you're going to get massive primer dimers. That means instead of annealing to your template, they're going to anneal to the other primer, right? You don't want that. So the way that you defeat that is you make your primers like that. Okay? That's the rationale for that. Let's see. Let me just go through these. I noted the stop codon. I needed that. Let's see. Change your resolution. How do you know what codon to choose? I showed you that. And that's that. It gives you another example here too, example 2. So when you design your own, you can use this document as an example for how you would do it. Okay, now let's talk about the protocol. So we've designed our primers. Okay, so the only thing to note, let's see, that's so annoying. One of the only things to note is your template. What the heck? Oh my gosh. Okay, your template. Point 1. How do you pipette 0.1 microliters? So this is of your plasmid template. How would you do that? Yeah, you'd make a dilution. So what you would do is if you had your Midiprep, you would take maybe 10 microliters from your Midiprep into a new tube plus 90 microliters of water. That's a 10x dilution. And then you would take 1 microliter of that. Does that make sense? So oftentimes if you see, pipette 0.1 microliter, nobody's saying actually pipette 0.1 microliter. Nobody does that. You make a dilution and then you pipette. The higher volume you use, the more accurate your pipetting technique is. So it's very, very inaccurate to try to pipette 0.1. You make a dilution and then you pipette 1 microliter of your dilution. Okay. Let's see. Another thing is, here's another question. Let's say he wanted to also mutate this codon. Can you do 2 at the same time? Yes. All you got to do is you got to change these 2 codons together. You change this, but then you got to move your primer over. Does that make sense? You would want to start your primer here to obey that 4 base pairs at least to the 5' end of your thing. If you were mutating this one as well, you would have to move this over. So I've done 3. I've done 3 codons at the same time. And it gets harder if they're spaced farther apart, right? Because if say you wanted to mutate this and this, that means your primers are going to have a lot more overlap and they're going to produce a lot more primer dimers. So that's going to decrease the efficiency. Okay. But 1 codon is super easy. And most of the time what we're doing is just 1 codon. If you need a whole section changed, that's when you move to like Gibson or recombineering or synthesizing, something like that. Quickchange is kind of only good for 1, 2, or 3 codons. Let's see, what else did I note? So same type of reaction. You just have two primers. You have a fusion buffer. You have DNTPs. You have a fusion polymerase. You have water. And then you have your template. And typically, sometimes, like I said before, sometimes it helps to have a hot start. So I will usually hot start quick changes. Here's the other thing to note, just like the Slim, the loops. So this is the looping of denature, anneal. I always set my anneal to 55. I never change that. Because again, I'm not worried about non-specific binding. I'm not worried about the specificity of my primers because my template is so pure. And then 72 is extension. And the looping, how many times does it loop back and forth? 25 times. And the rationale for that, same as Slim, which is what? Yeah, it's going to decrease the mutation rate because this is a technique that's going to PCR all the way around the backbone. So we want to limit the mutations that it's making in the backbone and in other contexts. Let's see, why such long extension time? Whoop, that's the final extension here, 3.5. That's pretty long. And again, that's because it needs to go all the way around the plasmid. Okay?
So whenever we do our project, we have to the temperatures for PCR.
I'm debating. No, because you won't change the annealing temperature of Quickchange. I always leave it at 55, regardless of what my primers are. All I need you guys to do is kind of get through the design. Once I know that you can design the primers, I know that anybody can, you're basically just cooking at this point. I know that anybody can follow a cooking protocol. You're just putting this much in a tube. So it's really the intellectual design of the primers, identifying the codons, that is really what you really need to practice. But that's a good question. Let's see. And then there is the DPN1 digest, which we talked about. So here's a good thing to talk about. You could do DPN1 for 1 hour, and people do that at 37, or you could do DPN1 overnight at 37. What do you think is going to be the differences in the data if you do 1 versus 2? Yeah. So I always do overnight. And what you'll notice is that, so if these are plates, let's say this is an amp, we're doing a quick change on an ampicillin plasmid, and we need to select and get rid of our template. If you do the DPN1 for 1 hour versus the DPN1 overnight, this one is going to have far fewer colonies, and this one is going to have a lot more. And that means there's going to be a lot more false positives that did not get your mutation because they're just taking in leftover backbone. Okay? And that means you're going to have to screen more plasmids with what technique would you use to verify quick change? We're not changing the insert. We're just changing, well, we are changing the insert, but we're not changing the size of the insert, right? We're just changing the codon. So how would you screen to make sure to see if your quick change worked or not?
Did you do one of the fish things?
Yeah, but that would be a pain. There's a lot easier way. Yeah, you just Sanger sequence it. Sanger sequence. Okay? You're changing the sequence. So you need to verify with the primer and Sanger sequencing of your things that your codon has been changed. So you Sanger sequence it. And usually what I'll do is if I do a quick change, you'll plate these out. I'll pick 3 colonies. I would grow them up in the LB amp, 5 mils. And then I would do the Midiprep, which you guys did in lab. And then I would Sanger sequence. And I would check all 3, 1, 2, 3, for my mutation. Okay? And the way that you can do that is actually really easy. All you need to do is you get your sequencing file. Your sequencing file will give you a FASTA format of sequence. Let's say it looks like, oh my God. Let's say it looks like this one at the top here. So you get a sequencing file, and if you pick 3 clones, you'll have 3 kind of things like this. And all you need to do is copy the primer you designed and control find for that sequence, right? Because it should now be changed. And if you find that sequence changed by control find in one of your Sanger sequencing reactions, then you know that it worked. And usually about 1 out of 3 will work if you're doing DPN1 overnight. Okay, so that's why I usually do 3, and you usually get 1 that worked. And then you would go back to that culture and you would make a glycerol stock to save that. I would fully sequence the whole gene then. If you didn't sequence the whole gene, if you just sequence this little region right here, I would then fully sequence the whole gene to make sure it's perfect, to make sure there's no mutations. And then I would database that strain and then do my experiments. Okay. So the problem is if you don't do the DPN1 if you just do it for a short time, you're going to have to screen more clones. It's going to be more work in the long run. So I usually will just set up a Quickchange in the morning, and then before I go home for work, I will set it up overnight, DPN1, and then in the morning, I do my transformation. Let's see. Okay. Transformation, we already talked about that. Midiprep, sequence analysis, we already talked about that. Documentation, I already talked about that. Okay. And then I would document, just a quick note, I would document the changes that you made you want to document that mutation. So if you did serine 691 to proline, you would document that. Okay, now let's finish the lecture by talking a little bit more about other mutagenesis strategies. So Quickchange is site directed, but there's other strategies where you're mutating things which are useful. So one of these is called directed evolution. Okay, so let's see. I'm trying to think of how I should best pose this. Let's pose a scenario. I thought about doing this experiment. I never did it yet, but I thought about it. Okay, so I study 2 proteins, SID-A and SID-B. And SID-B is that deubiquitinating toxin that causes sterility. That's the one that has the dub. Okay, and then there's SID-A, which is an antidote. Okay, so it's a toxin antidote system, and this antidote can rescue that dub activity. And these things bind each other. So SID-A makes a protein A, which binds to a protein B. And somehow this binding interaction is important for rescuing the sterility. So I wanted to study this conceptual system. And I wanted to understand, like, what are the residues that are important for binding to SID-B? So just like I said before, proteins are machines. A binding interaction, how could you imagine a binding interaction might come about?
They're attracted to each other.
Yeah, so maybe one has a lysine and an arginine, and the other one has, help me out here, what's it, D-glutamic acid or aspartic acid? Aspartic acid, and then let's say the other one, glutamic acid. These are negative. These are negative. These are positive. That's going to have an ionic interaction, right? So this could be a binding. This binding could be mediated by very, very specific amino acids. Okay, and I wanted to know. Now, this is just an example. It might not necessarily be those. It could be other amino acids. Could be hydrogen bonds. Could be other things like that. But when proteins bind each other, there's specific amino acids that mediate that interaction. So I wanted to know, what are the amino acids that mediate this interaction? But I don't know which ones to mutate, because I don't know which ones mediate the interaction. So I want to do a mutagenesis study to find which amino acids are important. Okay, one way to do this would be, I can measure rescue in yeast. So I can put SID-B in a yeast and it will kill it. And if I put SID-B plus SID-A in a yeast, it will survive. So I can measure whether things die or survive and whether this interaction is working together in a cell. So one way to do this would be, I could make random mutations in A and see if rescue is lost. Does that logic make sense? So the way that I would measure that is if things that normally would survive in the presence of SID-A stop surviving, I could measure that by making random mutations. I haven't talked about how to make random mutations yet, but I could do that. I could logically do that, okay? But this is actually not the best way to do it because I don't actually have a way to select for things that die. Like it's hard to study things that die because they're not alive. So if I take the reciprocal of this logic, there's a better way to do it. I could, let's say, here's another scenario. A1 and A2. In my system, it gets very, very complicated. There's many SID-A's, SID-A to the X, and there's many SID-B's, SID-B to the X. And these are very, very specific interactions where a SID-B-X only matches a SID-A-X. And so a SID-B-Y doesn't match a SID-B-X. Does that make sense? There's very, very specific lock and key type functionality. So imagine I did this. SID-B-1. Okay, so these ones rescue each other. SID-A-1 rescues SID-B-1. But I have a relationship where SID-A2, a different homolog, does not rescue. Okay? But I could make mutations in SID-A2 until it rescued SID-B1. And then I could sequence, and whatever changed was responsible for the rescue. And I could actually select for this. How would I select for this? Well, let me show you. If I take a yeast cell, let's say just an example 3 to the n. So 1, 2, 3, a million yeast cells. And in every single one of these yeast cells, I have SID-B1, SID-B1, SID-B1. And I also transform plus randomly mutated SID-A2. So this is SID-A2 with a mutation here. This is SID-A2 with a mutation here. This is SID-A2 with a mutation here. Different mutations that are randomly mutated. One of these might survive, right? Let's say this one survives. That one's going to grow on the plate, and then all I need to do is sequence whatever that was mutated in SID-A to figure out what changed. Okay, I'm running out of time here, but this is an experiment that would be called directed evolution. And I wish I had more time to tell you how you would actually do this, but let's just quick. How you would do this is you would do error-prone PCR. So I told you there's some cases where we want the polymerase to make mistakes. Directed evolution is one of those. So let's say I made 1 million different copies with Taq polymerase, which has a high error rate, and I mess up the magnesium. Let's say I mess up the buffer, and I put way too little or too much magnesium to mess up the Taq even more. So I increase the error rate to 1 in 100. Okay? That means when I amplify my SID-A, there's going to be, if it's 400 base pairs, there's going to be at least 4 mutations in SID-A. And then I can clone and make a library of a whole bunch of randomly mutated SID-As that I made with error-prone PCR, okay, into this plasmid X, and then that library is what I transform in this experiment. Does that make sense? Does it kind of just logically make sense? So I'm just basically saying that, and I'll end here. I'm basically saying that there's some cases in directed evolution when we want the error rate to be high if we can select for a phenotype that we could study. In this case, it would be I could select for rescue. And then we could identify amino acids that got mutated that are important for that interaction. So I'll end it there.